Thank you for attending this workshop on cleaning and disinfecting in a healthcare facility. We'll do our best to stay on track and get all the questions answered, as I'm conscious that it's only me and Andrew holding you back from your lunch. So we will get through this as quickly as we possibly can. As you can see, my name's Yvonne Taylor. I've been in the FM industry since 1995, where I started as a cleaning operative. I'm currently the Director of Cleaning at Mitie Healthcare, and a big supporter of the cleaning industry, an industry which I'm very passionate about. So I am a Board Director of the British Institute of Cleaning Science. I'm a Board Director of the CSSA, which is a Cleaning and Support Services Association. I'm the ex-chairman of the British Services Association Cleaning Committee. I received my chartered practitioner, the 12th person in the UK to receive that, in October of 2022. And I was one of the work stream leads for the rewriting of the National Standards of Healthcare Cleanliness 2021, along with some of my wonderful colleagues from around the facilities that are in this room today. So I'm very pleased to have with me, Dr. Andrew Kemp, who holds a PhD in disinfection and decontamination. Dr. Kemp is an independent researcher and considered a global expert in skin and surface disinfection. He's published over 20 research and educational pieces on various aspects of cleaning and disinfection. His book, The Art and Science of Cleaning, Disinfection and Disinfectants, was published in 2021 and well worth a read. He is the head of the Scientific Advisory Board for the British Institute of Cleaning Science, and he is also a UK antimicrobial resistant ambassador for the International AMR Insights Group, focusing on disinfectant-resistant bacteria. So what we're going to try and do today, or the aim of today, is to run this a bit like a fireside chat. That said, we're going to answer and have a conversation around some questions that were posed to us by the Mitie customers and colleagues. We asked them to tell us what the hot topics and questions were that they'd like to be answered in this session. So we'll try and focus on answering those as part of a Q&A. I'll leave this on the screen for a little longer. If you scan this QR code, it will automatically take you through to this presentation. So I'll put it on the screen now. It'll stay on for a little while, and I'll also put it back on at the end of this session. So, Dr. Kemp, welcome, and thank you for agreeing to join me in this discussion. We've previously had, in my opinion, some fantastic conversations and discussions around our topic today. But I was racking my brain to think of a question I could ask you that I'd never asked you, and I don't know why I've never asked you. But how did you move from biological warfare testing into infectious disease transmission?
Actually, that's probably one of the easiest things I've done in many times. For those of you, just to let you know about biological warfare testing. So I worked at Porton Down as part of the British military team there, looking into testing for bioweapons during the Cold War. So obviously, what we wanted to try and achieve was some fast, hyper-accurate testing to see if biological weapons were being deployed onto the battlefield. So that's really where my background and interest in microbial contamination of battlefield casualties. So it wasn't just simply protecting soldiers from the potential bioweapons that would be deployed onto a battlefield, but also if you were injured in a biological or chemical environment, obviously if you're going to require surgery, the last thing you need is a surgical team that has to have all of the PPE for a biological weapon. What you want is them into normal scrubs with normal operating garb so that they can get on and do the best surgery that they can do and hopefully save some lives. So it wasn't just a case of testing for the bioweapons out in the field, it was also testing for the bioweapons on patients coming into surgical facilities. So it was, and then that moved into the decontamination. So if you've got a positive result, how are we going to decontaminate a particularly penetrating injury? For those of you who aren't aware of sort of what weapons do nowadays and have done for some time, so when you get a penetrating injury like that, more often than not, if it's a high velocity round that's hit the casualty, there are at least 2 or 3 different areas of the body that round is going to go through. If it has if it's travelled through a biological atmosphere, how much of that bioweapon has it actually taken through with it into those 3 different areas of the body? So you know, quite a complex and quite a difficult task to do. So, you know, for me, moving from that into infectious diseases and cross-contamination, surgical site infection, which was, you know, really what I did in the health service for many years, was quite an easy transition. Obviously, you're dealing with a different situation. It's a lot less stressful, but it still has its stressors. You know, the reality is that it opened up a whole series of interesting fields for me to then go into. So actually, for me, it was quite an easy transition through from one to the other. I think the most difficult thing was actually finding funding and the time to do some of the research that I eventually did. But the movement from one to the other, they're very similar disciplines.
And did you specialise in anything when you switched over?
Ducking, I think was yeah. So testing was my passion. Is to find a way of very accurately, very quickly testing. If you're going to ask soldiers to remove PPE in a field, or you're going to ask surgeons to work on a surgical case, you've got to know that they're not putting themselves in danger by doing so. You know, there were many operational requirements that this solved. So soldiers, we knew that they were about 40% less effective when they were wearing chemical and biological PPE than without. So the sooner you can ask them to take them off and know that they're not going to die by doing so, the better. And for a surgical team, you know, operating in PPE is really very, very poor outcomes from surgery.
Okay.
So testing for that it was my passion. It obviously moved into disinfectants and decontamination and all that kind of stuff. You can't siloize those things.
Okay. So obviously, COVID for the cleaning industry brought up lots of new products that were coming in, wild and wonderful things that were coming in from around the world. But the biggest thing is whether it's persistent or non-persistent. What is the difference between persistent and non-persistent disinfection?
Right, okay, so everybody in the audience, I'm sure, will be, if you move on to the next slide, everybody into the audience will be aware of log reduction scale. So this is what we use to measure the effectiveness of disinfectants, really. To put it in a nutshell, is that it's not measuring apples against apples or oranges against oranges. It's apples and oranges. Persistent and non-persistent effect is very, very different over time. It's, I'm sorry for the audience if I sometimes appear quite cynical about the way things are done. then that's 40 years of experience of being cynical about how things are done. So I'm sorry if that comes across, but you know, it's just the way it is. Follow the money, I think they say. So if you are testing and using standard testing methodologies, you will test disinfectant log reduction scales at 30 seconds, 1 minute, and some go out to 2 minutes, and now in the US there's 5 minutes. Why? Because the chemical industry have known for years that that is about the length of time that their products will kill. So we've got contact time and we've got kill time. Contact time is the time at which a disinfectant is actually active and killing on a surface or the skin. Kill time is how long it takes for that disinfectant to actually kill a specific microorganism. So if we took something like E. coli, yeah, and we hit it with a bleach, then you know the contact time for the bleach is usually about 1.5 to 2 minutes. But some E. coli, particularly E. coli spores, may take 9 or 10 minutes of contact to actually kill them. So if that is the case, then you would need to have your cleaning staff go back and reapply the disinfectant to the surface, particularly the chlorides. You'd have to have them reapply that to the surface 4 or 5 times in one after another in order to make sure that you've killed the spores on the surface. So that's the non-persistent. So in 1972, I think it was, the first really persistent molecules were starting to be examined, and they were silanated quaternary compounds. We'd had quaternary ammonias for years, from about 1917, I think. But in 1972, Dow Corning produced a silanated version. In other words, it sticks and it stays around doing stuff. So from that point onwards, people have been trying to produce safe products that will stick to surfaces, stick to skin, and continue that log 5, log 4, log 3 effect, whatever it was that their original molecule would do, but do it over and over again. So what you've got on the screen is a log 5 kill for a non-persistent and a log 5 kill for a persistent, and what's the difference? So if I take 10 million bacteria, I'm sorry, you can read this for yourselves, but essentially 10 million bacteria, if I hit it with a non-persistent, after about 4 hours, I'm back up to about the place that I was. It's not a perfect science, because obviously it's very, very difficult to say whether or not a bacteria is actually capable of bifurcation or it isn't, how quickly it will bifurcate. You know, we did some testing on hand sanitizers, and just give you a for instance as to what can happen if you within this. So it's not a perfect science and never will be. So if I took you to the toilets now, I'd probably be arrested, but not given that. We wash your hands with soap and water, we dry them, and then we apply alcohol gel to one and not the other, and then I retest you in an hour. The hand that we just washed and dried with soap and water, and in comparison, the alcohol gel hand is 2.5 times more contaminated than the one that you just washed and dried. So when I put on a master's set to explore why that was done, they came back with really 3 reasons why that was happening. One was the gel itself is a high sugar. 2, when you wash your hands, the water runs over the skin and removes dead bacteria, etc., skin cells, etc., away from that. When you use a gel, it doesn't do that. You don't have that effect. It stays there. So dead bacteria stay within the residual of the gel. Once the alcohol is actually gone, which is 30 seconds to a minute, depending on the temperature of the room and the humidity in the room, again, not a perfect science. Once that's happened, the dead bacteria remain on the skin. Any bacteria you haven't killed now have a high sugar environment with lots and lots of dead bodies, which are protein and carbohydrate. So they go nuts. So bacteria that would normally reproduce and bifurcate every 20 to 25 minutes were actually bifurcating in 8 to 10 minutes because they were in such a great environment. It was nice, lovely temperature for them, all the environment, everything you could possibly want on the menu if you're a bacteria was there for you. So that's why, and that's what the master's students came back with. Those were the 3 reasons. So then you put a persistent, so something like a silanated quaternary onto the skin, and you do the same testing. Log 5 result with a decent silanated quaternary, and then what happens is every 5 minutes or so, it is doing that log 5 kill. So instead of after 4 hours, you've got the same level again. After 5 to 10 minutes, you should be close to 0. It never actually gets to 0. That's not quite true. I have tested certain products and they've got to 0. So in effect, properly disinfected, sterile surface. So you're all aware of the difference between disinfection and sterilization. That the only difference between the two with regard to how many bacteria or microbes are still alive, there's no difference there. But the difference with sterilization is you then take a further step in order to make sure that it doesn't recontaminate. Well, that's really, really difficult on surfaces, and we argued for years over whether or not these persistent molecules were actually sterilants or disinfectants. Why is that important? Because the regulators think it's important. What's important to me is what the outcome is. So I'm more interested in what the outcome is than what a regulator says, which box it ticks for what you can put onto a label. But the reality is, in my humble opinion, the persistent molecules, the persistent disinfectants, eventually, and it'd be people like you that cause that change, will become the predominant disinfectants in use. It's also much more difficult for bacteria to become resistant to disinfectants if you're using persistent disinfectants as opposed to non-persistent. And we'll move on to that later on in the
So obviously which product we use to disinfect can really play a big part in the cleaning activities that we do within healthcare. But as we've discussed previously, microorganisms are very clever and in protecting themselves, which brings me on to the next question about microbial resistance to disinfectants. And these links between disinfectant resistance and antibiotic resistance.
That's a really important one. So every year I go to Amsterdam and I present in front of 250 microbiologists, chemists, specialists in antimicrobial resistance. Of the 200, I think there's 215 global ambassadors that deal with antimicrobial resistance. I'm the only one that deals with disinfectant resistance. So every year I stand up and 249 people, their eyes go, oh God, he's on it again. And I keep saying, if you deal with antibiotic resistance and you don't deal with disinfectant resistance as well, then actually there is no point. You're spending billions, if not trillions of dollars, pounds, whatever currency, every year trying to solve antibiotic resistance. And if the resistance for that antibiotic is coming from the disinfectant use, then it's pointless. You need to do both. And every year, billions and trillions of dollars are spent on antibiotic resistance and 50 cents is comparatively spent on disinfectant resistance. It really is. Do you remember those ties where you had loads and loads of dogs on it and a cat in the middle? That's me. I'm the cat in the middle. They all just look at me and go, oh God, he's on it again. But the reality is the mechanisms which bacteria use to become resistant to antibiotics are the same mechanisms, well, at least 2 out of the 3, are the same mechanisms that are used for disinfectants. Bacteria have been around for billions of years. They're brilliant at being able to survive all sorts of different environments. They've survived ice ages, they've survived the previous global heating up, etc., far better than animals and mammals and mammalians, etc., have done. They are so good, and within their makeup, they have the ability to turn off and turn on mechanisms to protect themselves. And the reason that they don't have them active all of the time is because that's using up energy, and they're really, really good at using only the energy they need. So but they are predisposed to genetically available way of getting around and any kind of chemical that would affect them, whether that be an antibiotic chemical or whether it be a chemistry type disinfectant. So the particular one of interest to me is the efflux pump. I'm sure you're all aware of the efflux pump from antibiotic resistance, but actually the same efflux pump is used for chemical resistance. Now, obviously, viruses are slightly different. You mentioned the C word earlier, COVID, and the reality is that in order to gain energy, the energy that it needs, a virus will invade other cells. Viruses are brilliant at invading other cells. We can learn a lot from how they actually manage it. And in actual fact, in 1941, when we went, and the West went down the route of antibiotics, the East and the Russians and people like that went down the route of using viruses to kill bacteria because they can get through the bacterial wall. Yeah? So they're known as phages. And so they're very, very good at doing that. So bacteria have developed a way through spore formation, etc., through an extracellular membrane in certain types of bacteria to keep the chemical from getting to the internal mechanisms of the bacteria. And viruses actually pass through the wall of another cell in order to use the energy from the cell, the ATP within that cell, in order to be able to have the energy to reproduce. So getting a chemical to hit a virus, it's firstly got to go through the cell wall of whatever cell the virus has actually gone into. And when we go on to testing later, you'll see why viruses are so particularly difficult to test for. Because if they're in another cell, how do you get through and how do you determine whether or not it's in another cell or not? So we'll come on to that in a moment. So essentially, they are inextricably linked. You cannot disassociate antibiotic resistance and disinfectant resistance anymore. There are now bacteria circulating around, particularly in Southeast Asia, that are resistant to chlorhexidine, our first and most important skin prep, surgical skin prep, but also colistin, our last ditch, last hope antibiotic. So, you know, we need to deal with these things together. And what we don't know is whether or not the bacteria became resistant to the disinfectant and then the antibiotic, or vice versa. So it's 2024, and we don't know something as important as that. So if you don't take anything else away from today's session, it is actually we have been so poor historically in trying to test and trying to, and even looking for answers to these problems. We are still absolutely in our infancy of dealing with this.
Okay. I don't know whether you want to talk about any new discoveries that have come along? Or...
Yeah, I'll just mention this slide because it's up on the screen. There are lots of papers. So by scanning that QR code, you'll get this. There are lots and lots of papers that support what I've just said about antibiotic and disinfectant resistance. And, you know, they really are worth, if you're interested in this subject at all, they really are worth having a look at those papers. These are some of the great and good in disinfectant resistance research.
Okay. So just thinking about my previous life, and we've had a discussion around rotational cleaning. So I published a paper in 2020, August 2020, probably not the best time. And it was all around rotational cleaning because I did an awful lot of work with clean rooms in Malaysia, and there's no formal training for working in, even within the life science arena. So I created a training program specifically for that. And one thing that I did realise throughout it, especially in the pharmaceutical clean rooms, was that every week they changed their disinfectant. So the disinfectant was never the same. And yet I find it interesting that we use the same disinfectant every day. So actually, should we be rotating our disinfectants?
In my view, yes, you should. So the reality is that most people in the audience here will be using either a chlorine-based, a peroxide-based, or peracetic acid-based disinfectant throughout their hospitals, and they won't be rotating them. The reality is that once an efflux pump is activated within the cell of a bacteria, that will become disinfectant resistant to whatever has activated that particular mechanism within the cell. So the idea of rotating disinfectant, because they have different mechanisms of action on the cell wall and ways of getting into the cell. So those three main non-persistent disinfectant types have different mechanisms of getting through the cell wall and into the cell. Whether or not the efflux pump will then reject them and remove them, it depends on the bacteria. That's too broad a thing to give a single answer to. However, by doing that, by using those three non-persistents in rotation, so if you start with a chlorine-based, by the end of, say, a month, that you're starting to see a resistant strain become dominant within that whatever area you're cleaning, then by switching then to a peroxide, there's a chance that the bacteria won't recognize it, and so at least for a period of time until it becomes used to the peroxide, there's a chance that you'll be killing the resistant strain to the chlorine by using a peroxide. And then by switching again to peracetic acid, there's another chance of another way of getting through the cell wall as another way of actually killing the cell, and then come back again. It's not a perfect science. No. It hasn't been researched to anything like the degree it should be. Yeah. But as things stand today, that would be my best advice to anybody. The only other things I would say about the nuances about that are, don't try and do it on a 4-weekly. If you decide to do something like that, don't try and do it on a 4-weekly cycle. Do it on the first day of every month. Why? Because after about 4 or 5 months, you're changing over on a Tuesday. Well, you know, who does that? Oh, crikey, are we changing today or next week or whenever? If it's the first day of every month, some days you'll be using for 31 days, and some months you'll be using it for 28 days. So what? Is there any difference? At this moment in time, there simply isn't enough research evidence to say that the 3-day difference would make any difference whatsoever to it. And it's a punt. That is the best punt you can have at going at this, because we don't routinely test our surfaces. So, how are you knowing if a bacteria is starting to become resistant? I did some in a UK hospital in the southwest a while ago, where in the orthopedic operating rooms, the post-op infection rates for joint replacements were up significantly higher, at around 6%. And the orthopedic surgeons said, whoa, that's it. We're not operating anymore. Sort out this problem with the surgical site infections, or we're not doing joint replacements anymore. So the then head of infection control was at the end of his tether. We'd known each other for years, so he picked up the phone, rang me. I went down, did some testing, and we actually found that there were several pathogens in the operating theatres that were resistant to the chlorine-based products that they were using. So we simply changed that, and the infection rate dropped immediately.
Yeah.
Immediately. And so, the mechanism, how those bacteria, those surface and air bacteria were getting into the wounds in a UVC canopy, etc. That's, I think I wrote a paper on that.
It's definitely something that we're thinking about, and something definitely that does need researching, because we've been using the same disinfectants for years within the healthcare environment.
I think by using persistent disinfectants, because some things like 5th generation cyclots, there are no resistant strains ever reported to cyclots. 5th generation cyclots, not the first to 4, but 5th generation cyclots. There are no resistant strains reported at all, and they've been around for about 10 years now.
Yeah.
So that there's a good indication that actually they are the sort of way forward. Yeah. Okay. The hypochlorous as well. Hypochlorous acid is another one that, as far as I'm aware, there is no resistant bacteria to that either.
Okay. So you've mentioned testing quite a bit throughout this, and there is a number of ways of testing. There's some really simple solutions through to scientific ways of testing. So what are the best ways to test surfaces? So
there are multiple methods we use. Visual inspection, which is as a cleaning supervisor, cleaning operative, etc., does this surface look clean or not, is about as far as you can go at this moment in time, until something comes along that allows a cleaner or cleaning supervisor, that's the word I'm looking for, thank you, to simply do a test and see. So one of my biggest bugbears is total ATP tests. Absolutely useless, completely and utterly useless, should never be used for anything at all, especially in a hospital. The worst thing about an total ATP test, in my view, is that if you get a 0 result, because it only tests for extracellular ATP, adenosine triphosphate, so that's what they're testing for, but it only tests for extracellular. So if you've got intracellular ATP, which is in the bacteria, it isn't testing for that. And so it's absolutely no use whatsoever. A 0 result may mean that actually, yes, that surface is clean, we can do something on it, when in fact it isn't. And when I've done side-by-side testing with total ATP and bacteria-specific ATP tests, the
Poles
results
apart.
are poles apart.
Completely agree with you on ATP.
Yeah. The problem with culture and PCR, there are particular problems. Let's deal with one at a time. Culture. Culture was invented to take specimens from a human being. A tear, urine, stool sample, something like that, and produce a result that made you be able to look at you. So you grow enough bacteria to be able to look at. So there are several things that are a problem with that. Firstly, in order to see on a culture plate with the eye, you need about 1 million bacteria to produce something that you can see with the naked eye, give or take. Some are slightly larger than others, but about 1 million. So anywhere where there's 700,000 bacteria, you look at the culture plate, and there's nothing there. It's clear. So you get nothing grown on your culture. And there's 700,000 bacteria in that one spot. So that's one. The other thing is that actually surface bacteria and bacteria from inside the body like different environments. So inside the body, obviously, you take a culture plate and you stick it into a incubator, usually for 24, 36 hours and at 37 degrees, because that's what they're used to inside the body. That's their environment. When I culture from surfaces, I do it at between 21 and 23 degrees, because that's their normal sort of temperature within the room that they're in, especially in the hospital where the temperature is controlled probably better than anywhere else, or you hope anyway. Mm-hmm. So sticking a bacteria culture plate that from a surface into 37 degrees may kill the bacteria that you've sampled. So culture, it's about, if you talk to microbiologists, really good labs will culture about 35% of the available bacteria from a sample from within a human, what they were designed to do. It's probably less than 20% of bacteria that will actually culture from a swab taken from a surface. So that's really I mean, if I were to come to you tomorrow and say, right, great, we've invented this new test, and about 20% of what you're looking for is going to be shown. How long will it take you to kick me out the door? But that's what we have, and that's what we use. PCR, my biggest problem with PCR is it shows dead bacteria, dead viruses, as well as live. So if I'm using PCR to test this surface, and I take a pre-swab and I get 1,000, and then I hit it with a disinfectant, and I take a post-swab and it says 990, have I killed 10?
10.
Or have I killed all of them, but actually 990 still have enough DNA for the PCR machine to pick up? It is almost useless as a test for surface. So the metabolic test, a bacteria-specific rapid metabolic assay, is hyper accurate. You get a result within 2 or 3 minutes, depending on who's doing it. But it has its downsides. So it's good for bacterial numbers, but you can't tell species from it. So my recommendations currently are that you use a bacteria-specific rapid metabolic assay. I'll get new teeth next week, don't worry. You use a BSRMA, that's easier to say, to identify the areas of highest concentration of bacteria. So if I were to do this table here, I'd probably take 4 or 5 swabs, one from either corner, one from the center, look at the results for it. If I get 10 here, 200 there, 28,000 over there, that's where I take my swab for culture, because I'm much more likely to get a result from 28,000 than I am from 10. So it's a combination of the two. And if I'm looking to test for, if I'm looking to test for, I've got an outbreak, what's causing the outbreak? Obviously there's a human aspect to that. Is that around in the surfaces? Then I'd probably use PCR. So I'd use BSRMA to start, and then PCR for species identification. If I'm doing it to determine whether my disinfectants are working or not, then I would use a combination of BSRMA and culture.